does include these organelles, these myofibrils. And then we already met sarcoplasmic reticulum. So this mesh network that you see here, sarcoplasmic reticulum, remember this is a hollow organelle. So it is hollow on the inside. It has these manifolds, these enlargements there, and then connecting pipes in between, that kind of like cobweb material. But again, it's all hollow on the inside and it contains a lot of calcium. Now here's a very special structure, transverse tubule or T-tubule. Notice in the sandwich here, we've got this part, this very thick part of the sarcoplasmic reticulum. That's called the terminal cisternae. It's a very large manifold, a very large pipe. And then another set of sarcoplasmic reticulums here also has a large pipe, but in between is a transverse tubule. What is a T-tubule? T-tubule is known as, and you may make a note on the side somewhere, sarcolemma invagination. I wish I could. I can't really type it on this presentation.
I got you.
Kind of solid.
Forrest.
Sarcolemma invagination. We used that term before in the skin chapter to describe what structure? The hair follicle. Because the hair follicle is an invagination of the dermis and epidermis. Okay? So, being a sarcolemmic invagination, what happens is we go like this. So this is the surface, the lipid bilayer of the cell, the membrane, and then it does this little thing. And it really doesn't form a pocket like a hair follicle will. It actually has an opening, it's a tunnel from end to end. And on the other side of the muscle, it is open as well. And that's just a two-dimensional slice through a three-dimensional object. So realize these sarcolemmal tunnels are kind of going every which way across and within the cell. The entrances is here.
Sorry, Mike.
You see these little holes on the membrane? Okay? It says openings to transverse tubule, opening to T-tubule. So you go along the surface, going along that plasma membrane, the sarcolemma, and every once in a while you run into a hole, and you can dive in the hole, and you can end up on the other side. These holes lead to tunnels. The tunnels crisscross the interior of the cell. Why do we need that? Well, it turns out muscle signaling, the muscle impulse, travels over the surface of the muscle cell, right along the sarcolemma, or plasma membrane. We need to internalize that signal so that we can get the signal here. Okay, the sarcoplasmic reticula, which contain calcium, have to see the signal. And what they do is when they see the muscle impulse, they release calcium. Remember, they store the calcium. What's good about calcium? Just storing it if you can't release it, right? So to actuate the muscle contraction mechanism, you have to take that superficial impulse, you have to internalize it by way of these transverse tubules. And notice how the end pieces of the sarcoplasmic reticula are right up next to transverse tubule. Their interface contains special release channels, okay, what we call voltage-gated calcium channels, and they release calcium into the sarcoplasm, this area. Why is calcium so critical for muscle contraction? Well, it turns out we have to go deeper and look at the proteins that live here. We already talked about actin and myosin, but on top of those proteins are other proteins. They're called regulatory proteins. And when they're in play, they normally keep the muscle in a state of relaxation. So what we're doing with calcium, by releasing calcium, whenever these particular regulatory proteins see the calcium, they actually lift off the surface of the main proteins, particularly the actin, and then the actin and myosin filaments can actually touch each other so that contraction can begin. Okay? So there's a natural inhibition mechanism. We'll see it in an animation coming up. The inhibition says muscle under resting conditions does not contract. That is the default mode. We have to remove that inhibition in order for muscles to contract. And we remove it by way of calcium signaling. Does that make sense? Okay. So calcium really does not power muscle contraction. ATP, in fact, powers muscle contraction. But the presence of calcium permits muscle contraction. It's the permission slip for muscle contraction. All right? And then when all is said and done, you're done contracting, guess what you gotta do with the calcium that's in this? You gotta put it back where it came from, right? So there is a active transport mechanism that will shove the calcium back into the sarcoplasmic reticulum when contraction is over. All right, let's see what's actually going on inside. So here's a blow up of a myofibril. And can you make out the T-tubule here? And the flanking regions, what we call the, again, the terminal cisternae of the sarcoplasmic reticulum, just very large manifolds. Now this is where we're going to get a little bit into alphabet soup of structures. So here we have one pullout of a myofibril. That's kind of a three-dimensional drawing, more or less. But here's a simplification. So in 2 dimensions, we see a lot of banding appearance inside of the myofibrils. Okay. And it's actually this alternating light, dark banding pattern that gives us the striation so easily seen under the light microscope. So when you're seeing striated muscle tissue, you are looking at proteins, and these proteins are giving you light, dark. Okay. So what's going on here? Well, here we have a unit of a myofibril called a sarcomere. Sarcomere extends from here to here. So the actin and myosin filaments are kind of like parallel to each other, and they're in proximity to each other. So thick filament here, thin filament there. Now think about the thick filament, and we're going to see it in the animation. The thick filament has these heads that stick out, and they're always trying to touch the actin filament. So if this is the actin, this is myosin, the real situation, they'll show it in a better diagram, but they don't show it here. The head is like my fist, and it always wants to reach out and touch the actin filament. And what happens when they make contact, and if ATP is available, the head of the myosin filament nods. It kind of lurches forward like this. And these are all molecules, so it's a very small type of movement. But remember, there are many millions of these things, so the movement collectively can be very great. So here's what happens. When the filaments connect, flexion of the head, dragging that actin filament inward. That. Disconnect, attach at a different part of the actin filament, do it again. So it's a ratcheting kind of motion. Like that. And many of these heads contacting many actin filaments. So you see how the myosin filament in the middle here is kind of like sticking out on both sides. Okay? So the actin filaments on either side, they're saying tick, 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 bring it inward and draw it inward like that. Actin filaments are anchored on a special structure called the Z-line. Okay? And one Z-line to the next Z-line actually shortens the internal unit of what we call the sarcomere. And the myofibril shortens, the whole cell shortens. You get contraction. So the molecular mechanism behind muscle shortening or contraction deals with these filaments sliding past each other under the power of ATP. So that's what you're seeing. One sarcomere unit is an internal unit of a myofibril. It extends from one Z-line to the next. And there are many of these sarcomeres that go down the line to encompass the entire muscle. So sarcomeres are joined end to end, in other words. Now, I said earlier that regulatory proteins exist, okay? And they block the actin-myosin contact point. They actually sit on actin and cover its surface so that the two filaments cannot touch each other. And I said that's the default mode of muscle. So So in order to remove those inhibitory proteins, the calcium from here has to contact those proteins. And I may not have mentioned this in chemistry, but the reality is when molecules touch each other, shape change is often induced. Okay. Enzymes contracting substrate, some kind of shape change that results in some product. So calcium, it's as simple as Ca2 plus divalent cation floating around and touching the regulatory proteins will cause them to disassociate from the surface of actin and get out of the way. We're inhibiting the inhibitor. There's a special term for that in physiology. It's called disinhibition. By taking away the inhibition, you allow something to happen. So if this pipe were allowed to continue, again, that's the T-tubule, you would see it emanate on the surface somewhere. Other things that are in this alphabet soup, A-band, I-band. A-band is the dark band. Wherever myosin lines up, you can kind of make it out here. You see the myosin here? So wherever the myosin filament lines up are the limits of the A-band. A-band is the dark band, the dark striation that you see on this muscle. I-band is the light band. I-band is only where you can find the actin, and the actin is usually all by itself. Okay. With the Z line in the middle of the band. So all down the muscle, A-I, uninterrupted. Memory here is, so you can remember dark band. What's the second letter in dark? A. What's the second letter in light? I. So the A-I bands going down dark, light, dark, light. Now the region of overlap is still within the A-band, but even this H-zone region, which is central to the A-band, that's just where we have the myosin all by itself. You realize when we're pulling in the actin filament, there's a lot more overlap each time. So the central portion is just a portion where you find myosin. But anywhere myosin exists, so all of this is the A-band. Even the H-zone in the middle here, it's all A-band from there to there. H-zone is just an area of non-overlap. This dark area is where some actin is overlapping some of the myosin. Right? Now here they put in parentheses Z line or Z disc. The reason being is that this is a three-dimensional object. If you're looking at it on its side, take a frisbee, turn it on its side, it looks like a line, right? It's actually in 3 dimensions. So it's actually a disc structure. And that's the anchoring point for the actin filament. So everything gets better with an animation. And I think it's about time. Let's see, we'll take a break. Where's our break time? Oh, in about 5 minutes. So let's take a look at animation. Please work. Now let's take a look at this first one. This is user friendly.
You use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction, or remain unattached to allow the muscle to relax.
So notice that part of the video. ATP has to come in not only to cause the power stroke of contraction, ATP has to also come in to break the connection between actin and myosin. That'll be an important factor of discussion in a little bit. Now this part is going to talk about the regulatory proteins. I said regulatory proteins live on the surface of the thin filament of actin, and you can see them in play here. And they block the binding sites. So myosin wants to always contact actin as much as possible, but is prevented from doing so by these regulatory proteins living on the surface. They're occupying or covering all these binding sites. So we got to move them out of the way, right? And that's what we do with calcium. That's how calcium comes into play. So they're going to talk about what we call troponin and tropomyosin. Tropomyosin is this Twizzler strand of protein here, the longer strand. Troponin is a complex of 3 proteins here. What you need to know about troponin, it is the calcium sensor. So it's an adapter protein. It senses the calcium, it is connected to tropomyosin, and it moves the tropomyosin out of the way when calcium is present.
Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes. You use muscle.
Why does it have to revolve around note-taking? So not only that, but allow you to walk, to run, to lift things, to blink. It's a variety of activities.
So when you're having a muscle twitch, is that a chemical issue or a nervous issue?
Sometimes it is a skeletal muscle has been irritated by some kind of drug or toxin. Or it could be the nervous system that has been stimulated to release more neurotransmitter and kind of in an irregular kind of rate. So it all really depends. Now, let's take a look at we'll take a look at the second animation a little bit later. But you kind of get the sliding filament theory. Again, actin-myosin playing games with each other. And what is it, about time? It is about time. So let's come back at 20 after and finish up what we can with the muscle chapter.